இல்லை நான் ஸ்ட்ரீம் பண்ணுறேன். ஏன்னா பேக்ரவுண்டில் ஜிமீட் ஓப்பன் பண்ணால் தான் ஒன்ஸ் என்னோட கேமரா இந்த ஆஃப். இல்லை நான் ஸ்ட்ரீம் பண்ணுறேன். ஏன்னா பேக்ரவுண்டில் ஜிமீட் ஓப்பன் பண்ணால் தான் ஒன்ஸ்
மியூட் எடுங்க. மியூட் எடுங்க. சொல்லுங்க. மியூட் எடுத்துருமா? ஓகே. ஸ்ட்ரீம் ஆகல இல்லைம்மா? யூடியூப்ல இப்படிதான் இருக்கா? ஹலோ, மியூட் எடுத்துடணும். மியூட்ல இருக்கு வாய்ஸ்.
கேக்குதுங்களா?
ஓகே. சார்
ஓகே.
இப்படிதான் இருக்கா? ஹலோ, எதுக்கு மியூட் எடுத்துடணும்? என்ன கேட்கமா கேட்டு மியூட்ல இருக்கு வாய்ஸ். லைட் போட சொல்லலாமா? ஓகே. ஃபர்ஸ்ட் ஸ்லைடு போடுறீங்களா ஜெகன்?
ஒரே நிமிஷங்க.
ஃபர்ஸ்ட் டைட்டில் ஸ்லைடு மட்டும். ஹலோ, எதுக்கு மியூட் எடுத்துடணும்?
வருதுங்களா இப்போ?
குங்குமம் போட்டு மறக்கல போல இருக்குதுன்னா
அப்படியே தான் இருக்குங்க. ஆனா வருதுங்களா ஸ்லைட்ஸ்?
ஜெகன் இன்னும் வரல. டைட்டில் ஸ்லைடு மட்டும்.
வருதுங்களா இப்போ?
குங்குமம் போட்டு மறக்கல போல இருக்குதுன்னா
அப்படியே தான் இருக்குங்க. ஆனா வருதுங்களா ஸ்லைட்ஸ்? ஸ்லைட்ஸ் வருதுங்களா?
வரல. யார் பர்சன்? இப்போ ஓகே. இப்போ வந்தது. யாரோ ப்ரெசென்ட் பண்றாங்க.
சரி. दूसरे दिन दूसरा काम नहीं करना मैं उन्हें उठ के पता करता हूँ.
ओके.
வருதுங்களா ஸ்லைடு?
ஓகேமா. அப்படியே ஜூம் பண்ணு ஸ்லைடு. ஃபுல் ஸ்க்ரீன். ஓகே டன். அப்படியே இருக்கு. சாப்பிட்டீங்களா? இல்லை
இப்போதான் சாப்பிட்டேங்க.
சாப்பிட்டீங்களா?
வீட்டில் இருந்து எடுத்து வந்துறீங்க. கேண்டீன் போகிறது இல்லைங்க. அங்கே போய் இன்னும் கொஞ்சம் ரிஸ்க் ஜாஸ்திங்க. கேண்டீன் 5000 எம்ப்ளாய் பண்ணுங்க.
ஓகே.
அதில் சோசியல் டிஸ்டன்சிங்லாம் கொஞ்சம் காம்ப்ளிகேட்டடாங்க. அதனால இங்கேயே சாப்பிட்டுட்டேங்க. வீட்டிலேருந்து எடுத்துகிட்டு வந்துவிட என்னுடைய ஆஃபீஸ்லேயே சாப்பிட்ருவேன்.
சரி.
இப்போ தான் சாப்பிட்டேங்க. ஆக்சுவலாக 2 o'clock தான் கொடுத்துருந்தாங்க. இன்னும் 13-15 மினிட்ஸ் இருக்கு.
நோ ப்ராப்ளம். இப்போ செக் பண்ணிட்டோம். நீங்கள் எதனா சப்போஸ் வேலை இருந்தாலும் கிளிக் பண்ணிட்டு போயிட்டு
வீட்டிலேருந்து எடுத்துகிட்டு வந்துவிட என்னுடைய சரிங்க. மறுபடியும் ஒரு ஜாயின் பண்ணிக்கலாங்களா? அப்புறம்
ஒரு 10 நிமிஷம் போயிட்டு
சரிங்க. அப்படி விட்டுட்டுங்களா இல்லை 10 நிமிஷம் இருந்து வரட்டுங்களா?
இந்த ஸ்லைட் அப்படியே இருக்கட்டும்.
15 மினிட்ஸ் சரிங்க.
நோ ப்ராப்ளம். இப்போ செக் பண்ணிட்டோம்.
நீங்கள் பாய்.
வீடியோ மியூட் பண்ணிக்கோங்க.
சரிங்க. மறுபடியும் ஜாயின் பண்ணிக்கலாங்களா? அப்புறம்
ஒரு 10 நிமிஷம்
சரிங்க. அப்படி விட்டுட்டுங்களா இல்லை 10 நிமிஷம் இருந்து வரட்டுங்களா?
இந்த ஸ்லைட் அப்படியே இருக்கட்டும்.
சரிங்க. நீங்கள் பாய்.
வீடியோ மியூட் பண்ணிக்கோங்க.
कोविड सर्टिफिकेट घेतले सांगतात आणि आता कुठं असा होता
ஜூன்ல கூட ஒன்று கொடுக்குறேன்னு இருக்கு.
कोविड सर्टिफिकेट घेतले सांगतात आणि आता कुठं असा होता
ஜூன்ல கூட ஒன்று கொடுக்குறேன்னு இருக்கு.
नाही ना उलट ते म्हणजे की साधा आमच्याकडे आता काय फिल्म पण तिथे झालंय तर फिल्म सोमवारी काय आणि एकच सगळ्यात लॉन्च झालाय आज सोमवारी नाही आहे ज्यावेळेस ते म्हणतात काय तर सोमवारी ते कामाला काय लोक ऑनलाईन हे लोक आहे सोमवारी नाही आहे ज्यावेळेस ते म्हणतात काय केलं पाहिजे काम काढून घ्यायला पाहिजे काम काढून घ्यायला पाहिजे बरोबर घाबरलंय ते फक्त बस पुढे नवीन लग्न आहे ना घाबरलंय ते फक्त बस पुढे नवीन लग्न आहे ना मेडिकल बाजार काम करायचं त्यासाठी सांगितलं पाहिजे लोकांना काय नसताना मग काम करायचं त्यासाठी सांगितलं पाहिजे थोडस बेसल कुठे नाही अशी असेल त्याला ठीक आहे थोडस बेसल कुठे नाही किती असणार अशी असेल त्याला थोडस बेसल कुठे
Hello? Hello? Can you hear me? Hello? Hello?
Hello?
Hello?
Yes, yes.
Anna, yes. Can you hear me?
Hello?
Yes, yes, the voice is clear, Jagan.
Can you hear me? Good afternoon, sir.
Good afternoon.
Yes, yes.
Yes. Can you hear me?
Yes, the voice is clear, Jagan.
This is third session.
Good afternoon, sir.
Good afternoon.
Third session. Third second.
Can you hear me?
Trial session.
CM, kindly off your video, please.
Well, it's very good. Being the same, we can go to different places and come back to the trial session of the evening.
CM, kindly off your video.
We have learned Shastri is sitting at Mangalore and Sobaran is sitting in some place where I don't know which place she is. You are in Sobaran is sitting in some place where I don't know which place she is. This is last night. Good afternoon, sir. Good afternoon. We are waiting for Dr. Yero to join the program and it will take 1 hour starting the webinar
Hello.
we are waiting somewhere around 50 people have already
Hello.
So, credit goes to Saravathi and Pramod Kumar for this initiative. It is another 1 minute will be understanding concept the attendees should please be with your red line notebook so that a lot of the some of the performance will be without understanding very 1 or 2 minutes waited. We are going to start the live just waiting for us to react
All the participants, I need your all the participants, I need your
అరే ధ్రువాలు ఉందా ఏమన్నా సాంగ్స్ ఇంకేమున్నాయి అనిపిస్తాయా అరే ధ్రువాలు ఉందా సాంగ్స్ ఇంకేమున్నాయి అనిపిస్తాయా
okay.
Okay. 45 members వచ్చారు ఇంకొక 10-15 members వచ్చేంత వరకు వెయిట్ చేద్దాం
Okay.
All the
హలో
kindly mute yourself and do not present. It is disturbing the commencement of the webinar. Please mute yourself.
హలో
Kindly mute yourself and do not present yourself. It is disturbing the commencement of the webinar. Please mute yourself. All the participants kindly
హలో
mute yourself and do not present yourself. We are starting the webinar shortly. Please mute yourself. All the participants kindly mute yourself and do not present yourself. We are starting the webinar shortly. Please mute yourself. All the participants kindly we are starting.
హలో
Hello? 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 Hello?
All the term is archived by fact. A humble request to everybody: kindly mute your microphones and mute your cameras also, so that everybody can have a nice experience of listening to the speaker. And in the list of kindly mute your microphones and mute your presentation, so that you will be having everybody can have a nice experience of listening to the speaker. So let's start with the proceedings. And again, the savior action presentation, so that you will be having a meditation to protect themselves against COVID-19. We can't have a better timing to discuss the current topic and again current trends, actions, and vaccinology. On behalf of participants, the savior action, I welcome to K.S. Jagannathan, Deputy Director COVID-19. We can't have a better time to discuss the current topic and current trends, actions, and vaccinology. On behalf of participants, Hare Krishna, I welcome to K.S. Jagannathan and Deputy Director COVID-19. We can't have a introduction to today's speaker.
Video.
Actions and vaccinology. Hare Krishna, I welcome to K.S. Jagannathan and Deputy Director COVID-19. The introduction to today's speaker.
Video.
Hare Krishna, I welcome to K.S. Jagannathan and Deputy Director COVID-19. The introduction to today's speaker.
I am immensely glad and privileged to introduce the speaker of today's webinar, Dr. K.S. Jagannathan. Dr. K.S. Jagannathan is now serving as the Deputy Director and he heads the Production Department since November 2017 at Serum Institute of India Private Limited, Pune, focusing on formulation development, commercial manufacturing and troubleshooting activities for vaccines and vaccine adjuvants. Serum Institute of India Private Limited is now the world's largest vaccine manufacturer by number of doses produced and sold globally. Dr. K.S. Jagannathan obtained his PhD in Pharmaceutical Sciences under the guidance of Professor S.P. Vyas from Dr. H.S. Boud University, Sagar, India, in collaboration with Pasteur Institute of India, Konoor, and National Institute of Immunology, New Delhi. He did his postdoctoral studies at Tokyo University, Japan, and worked in collaboration with Konoor Pharmaceutical Limited, guidance of Professor Japan. Prior joining Serum Institute of India, he worked for Sanofi Group for about 14 years on various projects related to biologics, vaccines, monoclonal antibodies, and protein drug delivery. He also led the Formulation Innovation and Technology Group across Sanofi Pasteur sites, France, Canada, USA, and India. Dr. Jagannathan is a recipient of Best Scientist Award by Ministry of Science and Technology, Government of India, and was chosen as one of the key scientists to attend the Nobel Laureate meeting at Germany. He is also a recipient of several awards, and some of the key awards are BioAsia Innovation Award by Nobel Laureate Professor Martin Evans, UGC JRF, as well as AICT NDF. Some other awards, scientists, attending the Nobel Laureate meeting at Germany. like Young Scientist Award by DST, Government of India, JSPS, Postdoctoral Fellowship Award by Government of Japan, and Best Employee Award Platinum category by Sanofi Group of Company. He has several record publications in international journals besides oral presentations at international conferences and symposia as speaker. To his credit, he holds many US patents. He has more than 20 years of experience in the pharma industry at various roles in product development and commercial operations. So he is one of the most able to speak on the topic current trends in vaccines and vaccinology. And symposia as speaker. So we welcome for you to present your meeting. Many US patents. He has more than
Thank you so much. Can I start now?
yes, sir.
Okay.
We can start.
Thank you so much, and thank you so much for your nice introduction. And again, let me thank Dr. Hari sir. So he's known to me for more than 20 years. He was my senior, and he helped me a lot when I started my career in pharmacy. So I'm always grateful to him, and he is like my brother. So thank you, sir, again for calling me and giving me an option to present it. So let me start my today's presentation. So it's only global. We need to discuss, and if we have to go for a debate on this vaccines and vaccinology, I think it's like long one. So I will just start with like global view, and if you have any specific questions, feel free to contact me later as well. Okay, so today's my topic is current trends in vaccinology. And first, what is vaccine? I think everyone is aware. So vaccine saves life, preserve good health, and maintain high quality life. Vaccination stands as one of the most successful public health measures in the last century. So I will give you some couple of examples with some case studies how vaccination has started and where are we. And vaccine have provided some of the greatest success in the history of medicine, like eradication of polio, smallpox, and eradication of polio. So this has become like the most cost-effective, safe, powerful tool for medicine. If you look back, your hepatitis B vaccine, it used to be like 2,500 rupees per dose. Now we have for 50 rupees. So you see how the technology and cost-effective. So this has changed the world, completely, and it prevents, suffering, disability, and death from infectious disease. If you look back, so I just wanted to give some example pre-vaccine events. So birth defects caused by rubella. So you see, just imagine earlier, just to recap on this part. So children crippled by polio. Still we see some cases in India, like some our friends, like still a few of my friends have same earlier. I just wanted when they were young they had this issue. So still we keep seeing our friends. So horrific sound of baby struggling with fever, and neonatal mortality by tetanus, and rabies, that is a terrible skin of hydrophobia. Okay, so what are vaccines? So vaccines are material producing immune response and immune reaction. So immune response is nothing but immunity to natural microorganism. So it can be bacteria, it can be virus. So I will go in deep on each part actually. Vaccinology, this has expanded in the last two decades, and no longer it is a part of microbiology or immunology. So earlier it used to be part of microbiology. Now this has become separate, section. Prevention of infection in all age groups. So why have a vaccine? Every year up to 3 million children lives are saved by immunization. So you know very well, and everyone is aware about vaccines and immunization. So I'll just give you one example. So the disease case in US 20th century before advent of vaccines and after utilization of vaccines. So you will see the chart. So you have the column disease where you have different organisms, different disease. And the second one is estimated pre-vaccines, the cases before vaccination. So it used to be like in million, you see the case. Now the deaths in 2017, so some disease like polio and smallpox is almost zero, and other things are like very minimum. So you see the world like how it has changed. It used to be in million, Now the death in 2017 has almost become negligible. So this is the scenario, like how vaccination is now needed. So still some lobby, they still say vaccines are not needed, and this all fake, and you don't trust on it. So vaccine is the real, preventive medicine, and it helps you. You see, this table explains you. And what is vaccine? So classical vaccines can be weakened organism or dead organism. It is nothing but inactivated vaccines, or it can be individual proteins, or can be subunits. So vaccine is introduced into people to give a protective immune response that mimics natural infection, but it is not pathogenicity. Like it doesn't have any, it will not cause you any disease. So it is safe. So this is very important to understand what is vaccines. Okay, so when you go like type of immunization, so you have active immunization and passive immunization. So active immunization, it can be live attenuated vaccines like Rota, and a couple of COVID vaccines also is being developed. So I think one of the topic by my friend Dr. Dakshinamurthy spoke about COVID. So I will not cover more about COVID here. I will just give you global view on vaccines and vaccinology. So there are some inactivated or killed vaccines. So killed vaccines can be pertussis and inactivated poliovirus vaccine, and subunit vaccines can be tetanus toxoid, diphtheria toxoid, and toxoid based and recombinant hepatitis B based product. And recently for COVID, even many companies, they started working on DNA and mRNA based vaccines. There is no product till date in market, but though they started 10 years back, we didn't find any products in market. It has its own disadvantage, and I will just cover a little bit. So this DNA and mRNA based vaccines gives cell mediated immunity, but not humoral immunity, which is also very important. So I will cover a little bit about this, the immune response with some case studies in my later slides. So the passive immunization is nothing but antitoxin based preparation. So we have ready-made antibodies. So it can be either polyclonal or monoclonal antibodies. So if you take the tetanus toxin, so you have like a tetanus antitoxin. So when you have like any war, the soldiers are being administered with tetanus antitoxin, and you would have seen some products still in market like rabies. So we have both the antigen and antibody containing preparation. So whenever you give antibody containing preparation, it's called passive immunization. It is just basic. I will go in detail in my further slides. So comparative properties of vaccines. If it is live vaccines, what is live attenuated vaccine? So live, again, what is attenuated vaccine? Even yesterday I saw some debate on TV news. So they were not proper, like it was not a perfect definition. So live attenuated vaccine is nothing but weakened live organism by aging it or altering it, its growth conditions. So it replicates inside host, in the host. Attenuated in pathogenicity, it will not cause any disease. It may elicit broader response. Generally, live vaccines give broader response as compared to inactivated vaccines, and may require fewer doses and longer lasting protection. So it gives long-term immunity. And if you go to killed vaccines, it is unable to replicate in the host. It will not replicate in the host, and it cannot multiply or revert to pathogenicity, and less reactogenicity, and non-transmissible and more feasible technically, because no risk actually. Sometimes the live vaccines reverse back, and it's going to be disaster. So you need to ensure them in terms of safety and efficacy, and it's a little complicated procedures to demonstrate both efficacy and safety. Okay, so coming to subunit vaccines, it's contain only essential technically, because no risk actually. Sometimes the live vaccines reverse back, and it's going to be disaster. So you need to ensure them in terms of safety and efficacy, and it's a little complicated procedures to demonstrate both efficacy and safety. Okay, so coming to subunit vaccines, it's contain only essential technically, because no risk actually. Sometimes the live vaccines reverse back, and it's going to be disaster. So you need to ensure them in terms of safety and efficacy, and it's a little complicated procedures to demonstrate both efficacy and safety. Okay, so coming to subunit vaccines, it's contain only essential technically, because no risk actually. Sometimes the live vaccines reverse back, and it's going to be disaster. So we need to ensure them in terms of safety and efficacy, and it's a little complicated procedures to demonstrate both efficacy and safety. Okay, so coming to subunit vaccines, it's contain only essential technically. Yes, no risk actually. Sometimes the live vaccines reverse back, and it's going to be disaster. So we need to ensure them in terms of safety and efficacy, and it's a little complicated procedures to demonstrate both efficacy and safety. Okay, so coming to subunit vaccines, it's contain only essential technically. Yes, no risk actually. Sometimes the live vaccines reverse back, and it's going to be disaster. So we need to ensure them in terms of safety and efficacy, and it's a little complicated procedures to demonstrate both efficacy and safety. Okay, so coming to subunit vaccines, it's contain only essential. Maybe microorganism or winner man. It now it has come like you have combo vaccine. Earlier, if you take each disease, you have to take multiple injection. Now you have 6 or 7 antigens in one formulation. So you avoid 7 tricks. So you lot of companies are developing on combo vaccine, and cold chain management is very fixed actually. So we ensure 2 to 8, which is important for vaccines during manufacturing and also the shipment and immunization. So this is the technology has improved at the current stage. And this is a very rare picture. I think I wanted to share on. So this is nothing but actually Siena is a city located on the way from Rome to France. So it's based to be this city based to be most powerful economics of its time. So wealthy Incubians planned to build the largest cathedral in 1338. So after 10 years, so there was some plague outbreak in Northern Europe and killed more than 30% population in Northern Europe. So later it spread to Siena city, it's in Italy in 1348. And almost 2/3 of these wealthy individuals were died, and this left as such unfinished. So now this is considered as monument of infectious disease in Siena Cathedral. They started in 1348. So this is considered as monument infectious disease. So this is very important. So this has started even this type of outbreak happened even 1348, where no one knows about vaccines. Now we know what is vaccines. We understand science, and the situation is going to be different. But you see, even in 1348, 30% Europeans have died due to the outbreak of plague. Okay, so you should remember this is the trend of this outbreak even in 1348. So this is adult smallpox case. So earlier we used to have many cases. Now you have vaccine. This is almost eradicated. So we don't find such cases. And this all started with Louis Pasteur. So he has started the fundamental concept of attenuation. I think you are aware, you would have studied a little, the basic immunology. So I'm not going in detail about his invention. And introduction of aseptic techniques in medical plastics has improved now. And microbiology, immunology, vaccinology has become the separate section. And really we see the growth. So you see the lab when Louis Pasteur, when he was in his lab. So this is how it used to be. Now you see the facility, how it's how it has changed. So now, I just wanted to give one case study. Then I will go a little bit more in science, in how vaccines and what type of technologies how they work actually. So the polio vaccine. This is, I'll just give Indian scenario. So in 1985, you had around 1,50,000 cases reported in 1985. So it started decreasing. So Indian government has done fantastic job in reducing these cases by like oral polio vaccines, and now IPV is also in our government immunization schedule. So you in 1991 you had like 6,028 cases, and in 2009 it is 741. 2010 it is 42, and 2011 was one case. And the last case was 13 January 2011. Again, we never had any cases in India. So the last wild case of type 1 happened in 13 January 2011. So it's in West Bengal. And type 2 wild polio virus case happened on 10th October 1999 in UP. And type 3 happened on 22nd October 2010 in Jharkhand. So in India, like 13 January 2012 was a major milestone in the history of polio eradication. So we expect polio-free world by 2023. It's a tentative projection by different scientists and yet to be noticed. And I will just give you overview on Vaccine Institute in public sector in India. Like you have Afghan Institute in Mumbai, and you have Pasteur Institute in Kesoli, and in Kunnur, Uti, and you have CRA in Kesoli, again PCG Vaccine in Chennai. So it's all old, government organization. They have not upgraded to the current CGMP in all places except one or two in this list. So I will not discuss more about this part. And coming to the vaccines private sectors, you have many companies in India, and India is considered as one of the most, like in vaccine manufacturing all over world. India supplies more than 70% of vaccines goes from India, and we should be very proud on this part. And many companies from India, like you have Serum Institute. So we supply like we are one of the world #1 in terms of volume and number of doses sold. And you do have Santa Biotech now, it is part of Sanofi Pasteur Group, and Indian Immunologicals, Bharat Biotech, and Biological Events, and Panacea Biotech. So few companies supply few million doses, but we are in terms of volume, we still lead all over world. And coming to the immunization coverage in India, it is 2019. So still India has not, we don't have complete coverage. So we cover like only 46%. So still in terms of immunization, I think India still has to improve. And this is the immunization schedule which we follow in India. I'll not go in detail this part. I think everyone is having this part. This is the immunization schedule followed in US. So they have a couple of additional products which we don't have in India. Okay, so now I just gave you some helicopter view on vaccines and vaccinology. Now I will give you some, I will just want to share my experience in vaccines with some case studies. So when like this COVID outbreak started, now everyone is asking vaccine. So vaccine development is not easy program. I just give you ideal scenario. If you do not have anything, like basics of any disease, if outbreak happens like COVID, in ideal situation, if you have to develop vaccine respecting our regulatory guidelines, it takes 20 to 30 years. So you need to start with the development, then you have to show POC, then you will be in big companies, they have stage gate meeting, and you have to demonstrate feasibility, then clinical studies and regulatory clearance. It's going to be like long process. So in ideal situation, it is 20 to 30 years. And there are some product which we develop as like applied research. Say for example, now you have tetravalent vaccine. So you are moving, you want to change to pentavalent and hexavalent. So in this scenario, still we take 5 to 10 years time to develop and bring to market. So you demonstrate many things. The regulatory path in vaccines are complicated. You have written guidelines, and clinical phase only takes 2 to 3 years. So in this COVID outbreak, and all US or Indian regulatory bodies, they have simplified the path. And it really helps company to expedite or advance the product. So hopefully we are also working on a couple of product, candidate vaccines. So we see some results in early July. So if everything is successful, we may have, vaccine by end of this year. So I will keep you updated through your principal on this part. So we are also working on COVID vaccine. This is the ideal scenario. So you can't develop vaccine in 6 months, so which everyone is expecting. So the ideal scenario, it takes 20 to 30 years for COVID. But if you're even going for applied, research, it takes minimum 7 years. So now everyone, once you develop, you have the POC, then you need to make formulation. This vaccine has to go to different place, and shipment and the stability is very important. So that keeping a formulation, that product stable is really complicated. It is not like just cooking. So everyone thinks it's a complicated. So it needs a lot of science to make sure that protein, if it is a protein based or live virus, you have to keep them stable, so that you can be used with a shelf life of at least 24 months. So it leads not a formulation science, and you have to be very careful in formulating and ensuring efficacy, safety, and stability, which is very important for any vaccine formulations. So I'll just give examples of drug delivery systems that are very important in vaccines, which is going to be in future, milestone. And if you take conventional vaccines, even whatever vaccines we have or abroad, the WHO market or US FDA, so it's with aluminum adjuvants. So there are two aluminum adjuvants that are being used in vaccines. One is aluminum phosphate, aluminum hydroxide. So when you have this type of adjuvants, can we improve it? Can we have alternative adjuvant systems which can increase the, which can simplify the immunization and also the increase the protection? So I will just give you with two case studies. One is micro nanoparticle based system. So in particulate system, it can be microparticle or nanoparticle or dendrimers or soluble microneedles with nanoparticles. You have many options. So I think if you go in detail about each part, I think we need one class for each subject. So I will just give you overview and with one case study with micro nanoparticles. Then you have another option, vesicular system with the liposomes or neosomes. And what are the ideal requirements if you have to develop vaccine formulation? So as I said, the stability is very important. So it's ideal to have liquid formulation. And sometimes you have to go for lyophilizer formulations. Then again, COGM is very important. Like if the vaccine costs is like 15,000 rupees per dose, everyone cannot afford. So the cost of goods manufacturing should be kept in mind when you develop. So you see the process that will not add more COGM, unnecessary things can be avoided. So this is very important for the formulation scientists and industrial guys to consider COGM, since from development and selection of excipients is very important. Some excipients are banned in Japan, some are banned in EU, and some in India as well. So if you are entering in Japanese market, you have to consider what type of excipients should be considered for stabilizing your vaccines, and many parameters like excipients and physicochemical properties. And there are multi-base pre-formation studies to be performed before you choose your excipient. And easy operations, because when you develop, it's being developed by scientists, so he knows everything, and everything will be he can understand and he can do it little better. And everyone thinks it's a common sense, but common sense is not common to all. So you have to see and simplify the process. So later when you go for commercial stage, even the normal the technician should be in a position to manufacture without any failure. So your process has to be robust, and you should always consider lean principles. So this is very successful in Japan in automobiles, but in India we are still integrating in big companies like Serum. And scale up changes. When you work, like when you make batches at 1 liter, certainly it works well. When you go for commercial manufacturing, so we manufacture at 2,800 liters. So it's not easy. It is not just scale up. So you need to consider multi parameters to ensure vaccine stability. And the scale challenges have to be handled when you go for scale, commercial operations. Then additional characterization. Sometimes, for example, if you are successful in COVID, so we will be manufacturing in 3 facilities. So how are you going to ensure that the vaccine manufactured in facility 1, 2, and 3 are comparable? So you need to have some additional characterization in addition to pharmacopoeial requirement, and show that the products manufactured at different facilities are comparable. The physicochemical properties remain same, and of course, the safety, stability, and efficacy has to be ensured. analytical methods have to be available, and some vaccines still it's a challenge. So you need to have analytical methods. If you can have in vitro model, it is good. Sometimes we have to go for in vivo model and assume the case of like if you make 100 batches, and if you have hexavalent 6 antigen, if you have to go for in vivo, so how many facilities you should have like for only in vivo animal model? So it's these things have to be considered in keep in mind when you develop vaccines. And shelf life, always better to have more shelf life. At least it should have minimum 2 years is the ideal requirement. Some product we could more than 30 months. It is very important when you go for tender, and this vaccine should always have more shelf life. And also you should consider the market demand. If you manufacture and keep it, if it is not utilized, so you have to consider market demand, competitor and IPR issues when you develop your vaccines. If you have to survive in market, sustain in market for a long time. Okay, so I will just give examples with some studies of particulate system, which I spoke little a few minutes back. So the micro and nanoparticles, I said it can be microparticle and nanoparticle are dendrimers, and how to prepare. I think if you are in pharmaceutics field, you would have done some experiments. I will try to link with vaccines. So it can be prepared with some natural polymer like chitosan using either TPP, trisodium phosphate, or GSPS cross-linking agent. And you can also use synthetic polymer like PLGA, polylactic glycolic acid, PLA, PGA, or some nanoparticles. So there are, you have many options, many polymers, but all are not approved for human use by US FDA. So you have limited options to play with. Examples are PLGA. So there are some products with proteins in market, but there is no product in with PLGA based nanoparticles or microparticles with vaccines. So I will just give some case studies with the vaccines and its results. It's going to be of your interest. Okay, so this polymer should be approved for US FDA before we choose it. So I will give some vaccine candidate like hepatitis B with PLG as a polymer as a example. Then you have vesicular system, so it can be liposome or neosome. So there are some product like ambisome in market. So how to prefer? We have modified it to for vaccines. I will just give you with some case studies. So as I said, I will just present two part. One is a particular system, that is PLGA. So in PLGA, we have considered PLGA microparticles, and we have also modified PLGA, that is called modified PLGA, and we prepared some liposome in situ gelling system. So that can be used for vaccines. So our aim is to develop some adjuvants like microparticles or in situ gelling system, which can induce both humoral and cell-mediated immunity to give broad protection. And also the current vaccines are parenteral formulation. If we can develop non-invasive route, it's going to be it's going to be very important always to go for non-invasive route if possible, like nasal vaccines. So this carrier-based systems are considered for nasal vaccine delivery. So I will give you some results, interesting results. So before we go to next slide, let me give you like PLGA-based systems like microparticles. They all started like 25 years back. So they were not successful because low entrapment efficiency. So if you add 20 microgram, only 5 microgram gets entrapped. So because it gets exposed to organic solvent, integrity gets lost. So you will not have good entrapment efficiency. So if it is a recombinant based product, if you are wasting 80% components, it's not easy to manufacture and deliver as a vaccine. And risk of altering antigen integrity during formulation, as I said, this when you have a solvent, it deters the protein integrity, so that you will not be able to see that immune response. And also the PLG gets degraded and it leads to acidic environment inside PLG upon storage. So when you have this type of issues, it is not possible to have proteins in like PLGA microparticles, and you can't deliver them. So in this scenario, what we have done is we try to see the stability of hepatitis B, and we have worked with other protein model like HPV and other things. So they are confidential. I'm not presenting it. So I'll just show you hepatitis B, which was done a few years back. So this data will be interesting, and it's very important for viral based infections. Okay, so I will present two systems. One is PLGA, the another one is in situ gelling system. So as I said, our intention is to induce humoral and cell immunity. The current vaccine gives only humoral immunity, that is IgG. So cell immunity is not possible with the conventional vaccines. So when you go for this new adjuvants, it is possible to induce both the immunity. And we try to study the immune response. I will present in my next slides. So how we prepared is we have non-phase, that is PLGA with dissolved in dichloromethane, and we form primary emulsion with antigen and secondary emulsion later to form PLGA, microparticles. So this system, what we have done is we have used some stabilizers like Strigolos. We have explored more than 80 stabilizers and we have identified one stabilizer which was very good. And also we try to interrupt some antacid which neutralizes the acid during storage. So as I said, the PLJ gets degraded and leads to acidic environment. So neutralize the acidic environment. We interrupt some antacid which could neutralize the acid inside the PLJ microspheres to give better stability. So this is the PLJ microparticles. And what we have done, we also prepared in situ gelling system where phosphatidylcholine and cholesterol was dissolved in chloroform and methanol to form thin layer. Later we dehydrate with antigen with polyacrylic acid to form in situ gelling system. So this is, I just gave you like a pictorial with a glass flask. When you go for commercial, this is not the strategy. You need to consider other closed operations, which I am not presenting. So this is just for your understanding. We just prepared some pictorial form, and this is how we form in situ gelling system. And if you see the photo micrograph of both in situ gelling system and PLGA, so this is how it looks. And structural integrity, so you see the LAN1, this is nothing but SDS page with the silver staining, and LAN1 is plain hepatitis B before entrapment. You see this is actually it is integral. You can see there is no degradation. And modified PLG with trigolos, you see the LAN2, so it is intact even after formulation. So in situ gelling system also with hepatitis B, LAN3, it is also intact after formulation. Fourth LAN, you see you can't this is without stabilizer, you can see the degradation. So this is how the formulation, we could improve the formulation with stabilizers and antacid. So the protein integrity was okay. And we also ensured that we have done some additional tests like IEF, isoelectric focusing, Western blot, TEM, and relative potency, which I am not presenting here. I need 1 hour to talk on this part. So I'm giving something on characterization of both the in situ gelling system and modified PLGA. So the PLGA is negatively charged and modified PLGA is positively charged, and in situ gelling systems are negatively charged. So vaccines, whenever you develop, so we try to have like some nanoparticles, some microns at different size range. And what we realized is any vaccines with 1 to 10 micron gives maximum antibody response. If you try to decrease the size or increase the size, though vaccine is good, it is not giving good immunity. So the vaccine size is also very important to maintain between 1 to 10. So the developed formulations, all 3 formulations were found to be in within range, which we expect, that is 1 to 10 micron size. So entrapment efficiency, if you don't add stabilizer and antacid, it was less than 10%. So we could increase to 75%. So in situ gelling system, it was 5%. We increased to around 50%. So still it can be improved with further studies. So this is the compiled table to understand about the different properties. And I'll just give you link this data potential result with nozzle vaccines. So generally nozzle mucosa contains sialic acid which carries negative charge. So whenever you give negatively charged formulations, it gets cleared from the nose. So it is important to consider the surface charge in your formulation. So we modified the PLGA with using chitosan to have this positive charge, that is 27.2 is the zeta potential. And in situ gelling system, though it is negative, it was very good in inducing immunity. I will give you in my next slides, like that antibody response. So here is the nozzle clearance study. Lactose is the nozzle clearance study. So as a negative control, so when you take lactose, so more than 50% gets cleared within a short time. And when you go for PLJ, the blue one, second from the lowest part, so that is PLG negative charge. You see the clearance, it was faster as compared to other modified PLG and in situ gelling system. So the best mucosal clearance rate was with modified PLG because it was positively charged. So this is the nozzle clearance rate with gamma scintigraphy. So I try to plot in graph for easy understanding. And it is just pictorial explanation. If you have PLG particles with negative charge, and this is how when you take your vaccines, no mucoadhesion, so it gets cleared. It will not be presented to antigen presenting cells such as dendritic cells or macrophages. And if you go for modified PLG based microparticles, so you have surface charge which is positive charge. When you immunize, it gets mucoadhesion, then it gets antigen gets released and presented to antigen presenting cells to give immunity. So that just vaccines, when you change the surface charge, you see the impact. So previous in previous slide, there was no mucoadhesion, there was no antibody, and when you go for modified, just change this surface charge, you see the difference. So this is very important to consider multi-parameters when you develop a vaccine. And this is in situ gelling system. So you have in situ gelling system. So when you give, it forms a film. In situ gelling like film, then it transfers, it gives the antigen gets released and presented to antigen presenting cells to give good immunity. So this two system we have chosen for further studies, and we have ensured stability, and stability was okay up to 24 months. And I'll just give you some immune response data. Serum IgG, that is nothing but humoral immunity. So if you see this bar diagram, the first one is PLGA microspheres. As you see, this negative charge, and this is IgG. So you see how it responded. So it has given. But the second one is in situ gelling system, the ribosome-based in situ gelling system. And third one is modified PLGA, and the last one is conventional vaccines, that is aluminum. So here the conventional vaccine, as I said, it gives only humoral immunity, not cell-mediated immunity. So our systems which we developed, modified PLGA and ribosome based in situ gelling system, so both could induce IgG, which are comparable with aluminum-based vaccines which are in market. So if you go to next slide, IL-2 and IFN gamma, this is indirect marker for cellular immune response. You see the aluminum-based vaccines. So you don't find any, IL-2, the first graph is IL-2. So aluminum-based vaccines almost, very minimum immune IL-2. And PLGA, since it was negative charge, it got cleared from the nasal mucosa. We didn't find IL-2 response. And in situ gelling system and modified PLGA, you see the significant in quantity in terms of nanogram per spleen. And if you go to the second graph, that is IFN gamma. So you see the LM-based vaccine, that is conventional marketed vaccines. It has not given any IFN, and modified, PLGA, that is negative charge, also has not given enough response. But both modified PLG and in situ gelling system gave very good IFN level as compared to other two systems. So what I'm trying to say is, when you have a vaccines with the modified adjuvants like microparticles, it gives both humoral and cell-mediated immunity, which we observed with both in situ gelling system and modified gelling system, whereas the conventional vaccine gives only humoral, that is IgG. And since it's a mucosal vaccines, you have additional advantage. It gives secretory IgG in mucosal surface. Say for example, we have given through nasal route, and we see secretory IgA in all mucosal surfaces. See, for example, the first one is the violet color is LM-based hepatitis B, and the second one is PLGA, that is negative charge. It got cleared. We didn't find good IgA, and another brown and this light blue was with in situ gelling system and MPLGA, that is modified PLGA. You see the secretory IgA. So you see, when we immunize vaccine through one mucosal surface, it gives secretory IgG in all mucosal surface. Means, so entry of pathogen is through mucosal surface. So if you can develop non-invasive route like oral or nozzle. So it gives secretory in all mucosal surface, so which is entry for pathogen itself can be avoided. So this is going to be very good practical, good vaccines for HIV, where the transmission is going through mucosal route. Okay. So in short, what we have seen is we discussed about the global view on vaccines and vaccinology, and the developed systems are highly mucoadhesive. So it can be considered for mucosal vaccine. It's going to be practical in future, and high antigen loading we could achieve up to 75%, and antigen integrity was maintained up to 24 months. And it has got good stability, and we could induce both humoral as well as cell-mediated immunity, whereas the conventional vaccine gives only humoral immunity. So this is very important to induce both humoral and cellular, which is very important for viral infections. So I think I have 10 minutes for if any questions.
Yes, sir. Very good afternoon, sir.
Yeah.
Very good afternoon, sir. It was a very nice and excellent presentation, sir. I'm from Faculty of Dental Science, sir. Just beyond this virology vaccination, generally want to know about what are the vaccines currently available for dental caries?
Okay, I don't think any vaccines for this actually. So I don't know the market or something. If you want us to consider, maybe give us proposal. We would consider. So how much it's going to be useful, and is it really worth? Then we will consider it. So right now, I don't think neither my company or I have not seen any product, I believe.
Whereas in the case of most of the oral carcinoma cases, where we get are related with HPV, human papilloma viruses, sir.
Yes.
Can you suggest us some vaccinations, sir, regarding that?
Yes, actually HPV, you have two candidate vaccines. One is HPV L1 based and L2 based. There are some product in market like Merck, Cervarix and Gardasil by GSK, and even other companies. Even we are also going to launch. We have in phase 3 trial. So there are some product already in market like Gardasil, Cervarix are the vaccines.
Yes, sir. Thank you. Thank you so much, sir.
Any other questions?
Hello? Sir, can you hear me? Sir.
Yes, I can. Hello? Hello, I can't hear you.
Jagan?
Yes.
The voice is visible?
Anna, I can hear your voice.
Okay. Hello?
Yes.
There is a question.
Yes.
Dr. Jagan, there is a question.
Yeah.
Narayan Kirti. From Narayan Kirti.
Okay.
What do you know about dental carrier vaccine?
Sir, I'm the one who posted the question. I asked him directly only, so he had explained to me. Thank you. Thank you so much, sir.
Yep.
Okay, thank you.
Yeah. Thank you.
Any other questions?
The participants can raise their question or can type it in the chat box.
Hello, sir. Hari, sir.
Yeah. Yes.
Hello, sir. I am Professor Jay Kumari.
Okay.
We're from Wales University. Sir, any vaccines from plant?
Focus.
Phyto vaccines, sir. Any vaccines from plant? Any ideas, sir?
I think there were some vaccines under development. It was in phase 3, but I don't think some product in market at this stage. Actually, I have seen some companies even from Mumbai. So they were working, but plants again in microorganisms. Say for example, if you are expressing in E. coli, so we get one cell in like it gets multiplied in one day. So 20 minutes is the multiplication time, and we get two it gets increased in 20. But in plant, it is not going to be easy. So I have no idea about plant vaccines, but I'm sure there is no product in market.
Thank you, sir. Nice presentation, sir. Thank
Thank
you.
you.
Sir, Selvam, COVID-19 vaccine.
Sorry?
Hello,
Yes.
sir. What are the status of the COVID-19 vaccine?
Okay. So I will just give in serum there are more than 71 vaccine candidates in clinical trials, and some are in safety studies, some are in clinical one and clinical phase one and phase two. So we are working on a couple of candidate vaccine. One is there is a theory that BCG vaccine gives complete protection against different disease, including COVID. There was one publication and study report from New York University. So we do have recombinant BCG, which is improved version of existing BCG vaccine. So we are doing phase 3 trial, and we will have results soon. And we have collaboration with couple of other companies like Oxford University and Codanex, some other companies from USA. And so we are developing candidate vaccine against COVID-19 spike protein. So it is in clinical stage. So right now we don't have any results. So maybe we will keep you updated by June mid.
Yeah, great, sir. I hope the vaccine comes at the earliest and helps to save the humanity. And very
Yep.
good presentation.
Thank you.
Sir, one more question from Pawan Kumar.
Yeah.
Uh, these viruses actually undergo continuous mutation. And
Yes.
what would be the approach in developing vaccines towards them? Because even if you develop a vaccine, I mean there could be sudden change in mutation because of mutation. So Mr. Pawan Kumar has asked this question.
Okay, so we have a multi approach, the spike protein approach, which is very good, and the all probabilities are considered and linked with the missiles based carrier system. So we have collaboration with Codanex in US. So we have considered the all probabilities, and we expect it's going to be practical vaccine. So right now it is all in theory and hypothesis. So we try to cover all possible options. Let us hope for the best in clinicals.
Sir, which stage of mutation directly involved in the decrease of platelet count? I mean, asked by Mrs. Dhanalakshmi, sir.
Okay, so this is being led by different groups. So I am into product development. So I don't have right now exact answer. So this is being like, this is again vaccine is like multi group. So I am into product development. So once we get this candidate, then we convert to final product. So this part I have no idea. But if you need answer, I can find out with my colleague and I will let you know by mail.
Sure, sir. Thank you. I mean, so kind of you for doing that. Sir, a lot of acknowledgements and appreciations for the presentation flowing in continuously. And thanks for a very clear presentation, sir. And probably this is much needed, topic, to be discussed at this. Mrs. Gajalakshmi to present the vote of thanks.
Register my
Hello, Gajalakshmi ma'am is muted. Sorry, we couldn't hear you. Gajalakshmi ma'am is muted. Can you unmute her? Yeah, sure.
ஃபர்ஸ்ட்லேருந்து சொல்லுமா, இல்லை ஆஃப் வேலன்னு சொல்லு.
The webinar series organized by Faculty of Pharmacy, Dr. MGR Educational and Research Institute has started with the webinar. I would like to register my heartfelt thanks to Founder Chancellor, Honorable President, Honorable Secretary for all their support in all our efforts. With about 250 plus participants through Google Meet and 100 plus watching this live through live streaming of Google Zoom, I thank all the participants for their active participation. I thank our speaker, Dr. K.S. Jagannathan, Deputy Director, Serum Institute of India, Pune, for accepting our invitation and rendering an excellent presentation. I thank our principal, Dr. N. Harikrishnan, who stands behind all the efforts of Faculty of Pharmacy and leading us by example. I request all your support for forthcoming webinars also. Thank you, sir, with a lot of appreciation, clearly clarified our doubts. Question session still more informative with full of demonstration. Thank you, everyone, sir.
Hello. Thank you, Dr. Jagan. Thank you for accepting my invitation.
Okay, no problem.
Thank you very much. Really, your busy schedule, you allotted time and
no problem. No problem.
Thank you very much. I'll call you back. Okay.
Yes.
So
yes.
as per your the word, June also there is a session with you.
Sure.
Please. Thank you.
Yes. I will. Yes. Thank you so much. Bye.
Thank you very much. மெயில் ஐடி கரெக்டாக பண்ண சொல்லுங்க.
To all the dear participants, we will be receiving your feedback form very soon. Filling which with all the credentials in the right manner, we will be receiving the certificates within a few days.
Mail ID.
And we also request you to render your support continuously for all our forthcoming webinars. There is one more webinar which is scheduled on Tuesday, that is 26th of May, on the topic the role of Indian medicine upon Alzheimer's dementia. So I also, the flyer will be soon circulated. So I request everybody to kindly give their support for the forthcoming webinars. Thank you very much for joining us. That's all from our side today. Thank you.
Voice clear ಆಗಿತ್ತು. Sir, close.